Today we'll be discussing the gut tube and the body cavities. This will be a relatively quick lecture as there's only a few points that are pretty high yield that we need to cover today. So just a reminder that during the third and fourth week we have the process of neurulation. Now remember, at first the ectoderm is flat, right? You have flat ectoderm, but then it rolls and makes a tube, right? You have a neural tube being formed by the end of that process called neurulation. All right. So at the same time that neurulation is undergoing, you're having formation of the gut tube. So you sort of have a tube and then another tube. Okay. So you can sort of see that here. So you have this tube forming up top, the ectoderm, by the process we described earlier. And don't forget that the neural crest cells are going to be migrating during this stage. So all the stuff that we discussed at the same time, we need to consider what's going on with the endoderm. So there's neurulation and the neural tube. And now we focus here. This is going to be sort of the focus of the next few slides. The endoderm also starts to roll up into the gut tube. Now, the middle layer is the mesoderm, just as a reminder, and the mesoderm is going to hold the two zones together, this neural tube and this gut tube. Um, they're contained within, not contained within, but connected to each other by the mesoderm. And there's a visceral part of the mesoderm and there's a parietal part. The parietal part is going to form the body wall, and we'll see the implication of that later. Um, but the visceral part, again, viscera, eviscerated. When you're eviscerated, you lose your guts spill out. So the visceral mesoderm layer is going to surround the gut tube, and they're going to fuse. So notice here you have your parietal layer, just a review from last time. The parietal layer is going to come down, it's going to form this wall, right? But it's going to pinch off so that you have this forming here and this forming there. So these parts are continuous, forming the parietal mesoderm, the peritoneum of this is the more, I don't want to say this is the abdominal cavity, so to speak, but this will form the peritoneum. And over here is also parietal, formed by the parietal mesoderm. What I want to draw attention to is how this is parietal mesoderm, this is visceral mesoderm, and there's a part where they meet. So that area where they meet, it's going to be a double layer called the dorsal mesentery. So it's double layered. Why is the mesentery a double layered membrane? Because it is where the visceral mesoderm and the parietal mesoderm fuse. All right, and the dorsal mesentery extends from the caudal limit of the foregut, so bottom part, um, toward the end of the hindgut. All right, so that mesentery is really suspending almost all of the future GI tract. And when you do dissections, you're going to realize that when you dissect the GI tract out, it's all sort of still within its axis. So I just want you to keep that in the back of your head, how it's all arranged along the mesentery, and the mesentery comes from I don't want to say the fusion, but it's composed of both visceral and parietal mesoderm. So by the end of the fourth week, the lateral body wall has formed, as you can see. All right, the lateral body folds, have fused to form the ventral body wall. So really everything is tucked up, but we talked about that last time. So if the last slide was if this is my head and you sort of cut me this way and you look inside, this slide is if you rotate my head, slice me that way. So why don't you take a moment to pause the video and look at the previous slide and compare it to this one? Because this slide is if you took that same image that was like this, you rotated it this way and then sliced me through my nose to the back of my head. All right, that's going to form a sagittal section, whereas what was on the previous slide was a frontal section. Now, that means that this is the cranial end, this is the caudal end, this is the dorsal end, and this is the ventral end. And note how the ventral body wall is going to completely close off around the region of the umbilicus. All right? Look how this ventral body wall is closing off. Similarly, the midgut is going to communicate with the yolk sac through the vitelline duct. And this is very high yield. This is critically important. The midgut can communicate with the yolk sac through the vitelline duct, and that's normal until around the second to third month of gestation, where this should close off. The vitelline duct should degenerate, and failure of the vitelline duct to degenerate can lead to a Meckel diverticulum. All right? Failure of the not abdominal wall, failure of the vitelline duct to regress and degenerate is going to give rise to a Meckel diverticulum. And because, look at the lining of the endoderm, right? This is all the gut tube. This is all gut tube. And let's pretend that there's an outpouching here. A diverticulum is a pouch. So let's pretend there's a pouch right here. If I were to follow along the lining of the endoderm, notice that it's complete, it's uninterrupted. Okay, so the sac is going to contain that lining, this yellow lining that represents the endoderm. So it's going to be a true diverticulum, a true diverticulum, because because it contains, it's a it's a structure of the endoderm, and it just has failed to regress, and you have this outpouching. It's going to contain all of the wall, all the components of the wall of the gut. Specifically, um, it's different from a false diverticulum because a false diverticulum will not contain all the components of the gut wall. All right, so the Meckel diverticulum is a true diverticulum. It's derived from this lining here, and it's because the vitelline duct failed to degenerate, it's going to be a true outpouching that contains all of the layers. Why does it contain all of the layers? Because look at how the endoderm is going to be continuous with it. Hopefully you never forget this, because this is very, very, very important. Partially because examiners can contrast this with the formation of a false diverticulum. So there's a process called diverticulosis. And in diverticulosis, which can happen as part of normal aging, the pressures of peristalsis can cause outpouchings in the walls of the GI tract, all right, specifically the colon, all right? You can get colonic diverticula, okay? So I'm just drawing an outpouching here. Now the difference between this outpouching and the Meckel diverticulum is this was not a result of embryologic processes. This diverticulum formed because of pressure. So there are areas where vessels, and this is high yield, but you don't need to know it for your basic science coursework. You'll learn more about it in GI. There's an area where the vasa recta penetrates. So vasa recta are the vessels that come in a straight line, vasa recta, and they penetrate the gut wall. And there are nerves with it as well. But the point is, where the blood supply is coming in is more susceptible, and just take my word for it, is more susceptible to these pressures that are generated during peristalsis, such that outpouching can occur there. And because it's a mechanical process, it just represents gut wall being forced into a pouch. So that pouch won't contain all of the layers. It won't contain all of the gut layers. Whereas in the Meckel diverticulum, right, in the Meckel diverticulum, because it's coming from the endoderm, you're going to have all of the layers. All right, that's pretty important. You need to be able to distinguish between the Meckel diverticulum, which is a true diverticulum, and diverticulosis, which is a false diverticulum. And both of these can be an area of infection. So it can be a nidus, or a source in which an infection can form. It can be like a staging area for an infection. And Meckel diverticulum get infected, inflamed, etc. Diverticulosis, these false diverticula can get inflamed and infected as well. The main differentiation is how they form. All right. So between the 6th and 10th weeks of gestation, the contents of the bowels will actually leave the abdominal cavity to enter the umbilical cord, and then they come back. So that's called physiologic umbilical herniation. So they're going to leave around between the 6th to 10th weeks in that time frame, and they're supposed to come back. If they don't come back, the bowel stays trapped in here. And because it herniated through the umbilical cord, and the umbilical cord is covered by amnion, the gut contents are going to be covered by amnion. Okay, you have all this amniotic lining. Now, you can screen for this defect with elevated alpha fetoprotein. So that's one of the things you can check in mom in the amniotic fluid to see if this issue is there, because there's going to be elevated alpha fetoprotein. But that's not too important at this point. What I want to emphasize is the fact that this is going to be covered by amnion. Why? And why might examiners test it? Because it represents a physiologic process that can go wrong. So the bowels are going to herniate between the 6th to 10th weeks of gestation, and then they're going to come back out. And then if they don't come back out and they stay trapped in there, they're going to be covered by amnion because the umbilical cord is covered by amnion, and it's going to form this sac called an omphalocele. All right, an omphalocele. And that's going to be different from gastroschisis. That's going to be different because in gastroschisis, and I'm probably butchering the pronunciation, the bowel is going to herniate through a defect in the abdominal wall. So let's say a defect is here, here, etc. Um, there's going to be an opening in the abdominal wall that the bowels herniate through. And when they herniate through that, they're not going through the umbilical cord. They will not be surrounded by amnion. Okay, and we'll see that on the next slide. So don't worry about it too much for now. And what I want to point out on this slide is gastroschisis. And you can see here, there's an umbilical cord, there's herniated bowel, but this herniated bowel doesn't have an amniotic covering on it. All right, and that's important. That's going to be a distinction that allows you to separate it from the omphalocele. The omphalocele is an umbilical lesion, and it has amnion covering it. This is adjacent to the umbilicus to the right, and it does not have amnion covering it. This is Ectopia cordis. Not super high yield, but see how these parietal folds are going to come in? There's a pericardial cavity here that contains the heart. If these folds don't completely fuse, so imagine that this is going to sit outside of the cavity. All right, so it's not going to be inside this cavity if it doesn't fold properly. Not very high yield. Don't stress too much about it. This is bladder exystrophy, and this is cloacal exystrophy. Um, don't stress out too much about these things. I wouldn't worry about them at this point. More importantly, be able to differentiate gastroschisis from omphalocele for the purpose of this lecture. There's not a lot that's high yield here. Just I want you to note the septum transversum, how it's folding um, right below the heart, and how there's these communicating areas called the pericardioperitoneal canals. Okay, so the peritoneal cavity or the abdominal cavity is sort of still communicating with the primitive thoracic cavity, and they communicate through the pericardioperitoneal canals, and that won't stop until the diaphragm is formed. And the septum transversum, which you can see sitting here, is going to be the scaffold on which we build the diaphragm. All right. There's not much that's high yield that I want to point out here. Um, really, just the fact that these pleuropericardial membrane folds are going to grow together, and that's going to form this area around the heart, and the phrenic nerve is going to be inside. And the only reason why I want to point out that the phrenic nerve is inside is because if you go into the plane of the heart, right? If you go this way inside, into the plane of the heart, into the plane of the screen, such that you're going in like this, what's below it is going to be the septum transversum, which is going to be a scaffold for the diaphragm. So I just want you to note the embryological anatomic relationship between the phrenic nerve and the diaphragm. That's why, part of why C3, C4, C5 keeps the diaphragm alive through their contributions to the phrenic nerve. I will talk more about why those somites, not those somites, but why those cervical nerves are the ones that innervate the phrenic nerve to innervate the diaphragm. All right. And over here, you have the pleuroperitoneal folds. So notice how they're projecting in. All right. They're projecting in and they're taking up the space of the pericardioperitoneal canal. And remember, the pericardioperitoneal canal was the only way that the thoracic cavity and the abdominal cavity were communicating. And now it's going to be closed off. Okay. By the 7th week, these folds will become more thinner and there'll be these pleuroperitoneal membranes. So these folds will thin out and there'll be membranes. They'll form these membranes and they'll grow medially inwards. Okay. Notice how they're growing in medially to form, to fuse with the mesentery of the esophagus. Okay. And that's going to form part of the diaphragm. All right. Now, they're going to keep on growing and they're going to keep on growing. And notice that they're starting to cover the septum transversum. So they're growing in here and they're starting to surround the aorta. And that's going to form the crew of the diaphragm. But don't worry about that right now. Not high yields and not important at the moment. The pleuroperitoneal folds are going to grow in, they're going to grow in, and they're going to surround. And then eventually they're going to start coming down into the septum transversum. Okay, they're going to cover the septum transversum until they're fusing into each other. See how they're coming in together? They're going to fuse into each other. And they're going to completely cover the septum transversum. So I want you to reimagine you had your septum transversum with your pericardioperitoneal channels. Now imagine these pleuroperitoneal folds are growing and blocking out these channels and then coming together to cover the septum transversum. Now, where they fuse over here is going to be called the central tendon of the diaphragm. All right, this is the main tendon for the diaphragm. And this peripheral area that they form, the peripheral area, is going to be connective tissue that is a scaffold for bringing in migrating myoblasts, which are going to give rise to more of the diaphragm. So when we say septum transversum is the scaffold on which the diaphragm is built, this is why, because the pleuroperitoneal membranes are going to fuse and recruit, what do you call it? And recruit myoblasts. Now these muscle cells, these myoblasts are coming from the cervical segments 3 to 5 of the cell mite. My apologies, give me a second. Sorry about that, I had to pause to clear my throat. So the innervation of the diaphragm is going to come from what? I'm going to let you try to guess it. From the phrenic nerves. Why are they coming from the phrenic nerves? Because the phrenic nerves are derived from the ventral primary rami of spinal nerves that were carrying the motor and sensory fibers that originated from C3 to C5. So the phrenic nerve is coming from somites C3 to C5, innervates the products of C3 to C5, and that includes the diaphragm, because the myoblasts are going to migrate. Remember that process that we talked about? Okay. And here I just want to show you what the diaphragm is going to look like. So here we have an opening for the superior vena cava. Notice how here there's the superior vena cava, and you have the esophagus opening, esophageal hiatus. Notice here, this is the gut tube, this yellow endoderm right here. So notice that it keeps its embryonic configuration, right? Where superior vena cava was higher than the endoderm, and the endoderm was higher than the aorta. So that's why the aortic hiatus is lower. The esophageal hiatus is a bit higher than that, and the superior vena cava hiatus is going to be the most superior. Okay, so you don't have to memorize that it goes superior vena cava, esophagus, aorta in terms of where the openings are in the diaphragm. You can remember it because of this configuration. So if the pleuroperitoneal membrane is going to provide with the septum transversum is going to provide the scaffold on which the myoblasts are going to migrate, the myofibroblasts are going to migrate to, then absence of that membrane means the myofibroblasts can't get there. Consequently, maybe there is pleuroperitoneal membrane, but the myofibroblasts just fail to migrate and fail to populate that area. What the implication of that is you're going to have a defect in the diaphragm. And this defect in the diaphragm is relatively high yield. It's called a diaphragmatic hernia. And it's very important that you can recognize this on imaging. All right. Notice here there's a normal right lung, but over here there's a tiny left lung, and this is most commonly a left-sided process. But the left lung is underdeveloped. Why? Because the lungs have to expand. That's why I was emphasizing that. The lungs have to expand to grow. The lung bud has to expand to grow. But if there's guts in there, and the guts herniate, and there are guts in there, there's no room for it to grow. So the lung stops developing, and that's called lung hypoplasia. Hypoplasia, less growth. Plasia means growth, and hypo means less. You get lung hypoplasia. And on imaging, I pulled this image off of Radiopedia. First, let me tell you what I'm looking at. This is a chest x-ray, and some of you might be able to recognize bones, and there's softer, less dense tissue over here. The darker tissue is, the less dense it is. And by darker, I mean black, like this. So notice here is air, this is air. How do I know that this is guts that have herniated? Well, what's inside guts? Just a bunch of gas. Guts are gassy. Okay, so I look here and I see a bunch of structures that are sort of like tubes, right? Like a lining and a lining and a gas inside and gas, 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 gas. But there's like organs. It's not that it's just pure dark. So there's lung hypoplasia for a different reason, but I can see outlines of tissue, and within these outlines is gas. Okay, that is diaphragmatic hernia. That is bowel inside the thoracic cavity, and the lung is not able to grow due to the presence of this. This is very high yield, and unfortunately, there's not much you can do for the baby. It's usually lethal. Now, I'm going to re-emphasize this occurs because of a failure of myofibroblasts to populate, or if the pleuroperitoneal membrane never existed. So either the fibroblasts didn't populate it or the membrane never existed in the first place. More commonly, it's because the myofibroblasts never migrated. All right. So that's the end of this lecture, and hopefully this was much more palatable than the other ones. Um, and this might be the last of the embryology series, but I have yet to decide that. All right, take care.